Good afternoon and welcome. I am so excited that you have chosen to join us today for the Van Andel Institute Public Lecture Series. We have an exciting conversation for you today. We're going to answer some big questions. For instance, people who should have genetic diseases and don't. Why is that? What makes them unique and special? And what can we learn about that and harness it to better treat those diseases? We'll also take a look at what might be in your very own backyard that could help provide future treatments. I am so excited because today we have Dr. Nick Burton, who is with Van Andel Institute, joining us to lead us in this conversation. Welcome, Dr. Burton.
All right. Well, thank you for that introduction, and hopefully we'll answer all of those questions, or as much as I can, today. So as Miranda mentioned, I am interested in people who should have genetic disease but don't. And so I'm going to lead as an example of what I'm talking about here with a story that was in the New York Times just a couple of weeks ago. So some people on this call might have read it. And this is the story of Doug Whitney. Doug Whitney is an individual who comes from a family who has this variant that should develop Alzheimer's disease. His mother, his brother, 9 out of 13 siblings, and many members of his family going back generations have developed Alzheimer's in their 40s or 50s and succumbed to disease. But Doug Whitney is 76 and shows essentially no signs of Alzheimer's disease. To date, he's the only known person to have this variant but not have Alzheimer's disease. And so this came out this year in a Nature Medicine study, and they profiled Doug Whitney. They've been studying him for years, trying to figure out what protects him from this disease, but the answer is still unknown. And the New York Times article ends with this quote that the puzzle that protects Doug Whitney is too valuable not to answer. I can't quite see the exact quote here, but this, I think, is really the thing that I'm most interested in. I'm fascinated by these stories because you'll see a story like this every couple of months almost. This is the story of Doug Whitney, and so it's in the New York Times because Alzheimer's is a very prevalent disease, but you also see examples of this in other contexts. So last year, there was a small study in France. So this is a hospital in Dijon, France. And there's a cluster of 5 university excellence hospitals there, and they predominantly take in children with genetic diseases, and they'll sequence their genome and try and figure out, what exactly is causing the symptoms these children have. And then they hope that they can, by assigning it to a specific syndrome, give them better, treatment or care for that pathology. And this is pretty much standard of care. But what this hospital did is they also decided to sequence the parents. And this was a bit non-traditional because for all of the ones that have been identified that they were working on, there are no examples of anyone who has these variants and doesn't have the disease. So they're usually thought to be essentially completely penetrant, and they just sort of arise by chance, but the parents shouldn't have them. And what they found is 12 different cases spanning at least 9 severe neurodevelopmental syndromes where one of the parents would have the exact same variant. They passed it on, but the parent is asymptomatic. They have absolutely no signs of disease. And in 9 of 12 of the cases, they were the first ever individual to have one of these variants, but not have the disease. So a lot like Doug Whitney, right? Where they have their genome looks like it should have a genetic disease, but they are otherwise asymptomatic. And this is true across many, many pathologies. A few labs, because these days we've started to study this at scale, or a couple of labs have tried to do this. You don't have to know anything about these exact studies that you see here, but basically we have a lot of human genomes these days, including genomes from otherwise healthy individuals. And what people are starting to notice is that approximately, there are different estimates ranging from 1 in 20 up to 1 in 20 to a little bit more rare, but it's not uncommon to find individuals who should have a given genetic disorder, but don't. They present as healthy anyway, and this is relatively common. And so my lab is interested in why. Can we figure out what's going on here to try and develop better treatments for these currently untreatable diseases? And so I'm just going to keep going on here. These are basically broken down. If I go back to that blue slide, as what could be going on. And there's really three theories, right? One is for people who definitely have this mutation where they validate all the artificial artifacts, it's genetic suppression, incomplete penetrance, and the environment. And I'm going to walk through what those are because they sound like very technical terms, and they are, but I think we can explain them in a way that will make sense to everyone. And so first off is genetic suppression. And so the first thing I'd like to highlight with genetic suppression is a model of like an apartment complex. Like if you think of an apartment complex and the plans that make up the apartment complex are your DNA. If you have a change in your DNA that breaks something, like let's say it breaks the fire alarms in a way that sets off the sprinklers, even though there's no fire, right? This is an immediate problem. This outcome is bad because you're flooding the building, right? So that's a problem that's in your, let's say a change in DNA causes the fire alarms to go off and now you're flooding. But let's say you had a second variant, right? So if I go here and click my slides over, there we go. Let's say you had two changes in the DNA. One that should set off the sprinklers, but another one that blocks the water pipes leading to the sprinklers. Now you've broken two things. You both have the fire alarm going or trying to set off the sprinklers, but also no water coming to the sprinklers. From the perspective of your apartment complex, you do have two broken things, but it's kind of fine, right? As long as there's no fire, the apartment complex is fine. That is genetic suppression. Basically, things are pretty much fine, even though you have two breaks. So you can have this in a genome where one thing breaks, it should cause pathology, but you break another thing and things kind of balance out. And this is actually how a lot of drugs sort of in principle work. For example, I have injured my shoulder and I have a shoulder and it has an injury. And that's bad. That's not a DNA problem. It's just like I have a shoulder injury and it hurts. And I'll take ibuprofen. And now that ibuprofen actually just sort of inhibits the inflammatory response. And then feel fine, right? And so I feel fine, but I still have an injured shoulder and I've blocked part of my immune system, but it's like more or less fine because the system is going to be fine. And so that's kind of the principle of a lot of drugs and genetic suppression is that you can turn other things off to make the system fine. The problem's still there, but it doesn't cause any bigger problems. And so genetic suppression is one example. Maybe these people have another variation in their genome that protects them from whatever that first variation was. The next example is incomplete penetrance. Incomplete penetrance is usually pretty hard because it's very hand-wavy, even amongst scientists. But let's picture something very stochastic. Like if you had a DNA change that instead of setting off the fire alarms was like in the foundation. And so the building just like falls down, right? That's a very chaotic process. Everything sort of just falls down. And that's obviously bad. But when buildings fall down, like occasionally they'll fall down in such a way that you'll get a pocket where there's like a room that's unaffected. This happens, it's stochastic. There's no real cause for it. It's just chance, right? Just lucky chance, and stochastic processes in biology might lead to some individuals just ultimately not getting disease because there's a bunch of noise in the system. That's the best way to explain incomplete penetrance without going too deep into the details, is that it's just noisy. And so some individuals are accidentally healthy. This would be the worst outcome for us because there's no way to make that happen in a given individual. But we don't actually think that that's what's going on here. And so the third example is the environment. And this is often pretty hand-wavy too, because it could be anything, right? The environment is anything. But what my lab posits is that most humans live in environments that on many scales are similar. Like the oxygen concentration we breathe is more or less the same, right? We all live on Earth. But what is variable is your diet and a microbiome. This actually varies quite a bit from person to person, and I'm going to go into a little bit more detail on this, but that those differences in diet and microbiome sometimes might explain some of these cases where people become more or less healthy based on whatever interaction with their genetics that they have. So your genetics interacts with its environment, and it can give rise to different outcomes. And so maybe some of it's environmental. This is the most exciting aspect of it for me from a therapeutic standpoint, because the environment is a place where we can intervene, right? We can do something about that for patients with a given genetic background. And so I'm going to go a little bit more in detail on this. So what is diet? when most people hear diet in media or in texts or in books, they're thinking of something like these two pictures on the left, right? Diet is like a pile of foods that we associate with health, or a pile of foods that we don't associate with health. That's, for the purpose of this talk, not what I'm talking about. Because from my perspective, or from the perspective of biology, of what's going to matter is the molecules, so the individual molecules. From the standpoint of your gut, that's what it is picking up and taking for food. The molecules that make up what you're seeing there as healthy and unhealthy, lots of them are going to be the same, right? It's just like proteins and nucleic acids, like just basic biological building blocks. But there also are differences. Also, diet is not one thing in the sense of, like, let's say I have 2 pieces of celery. We think celery is a vegetable, it's a diet. But if you grow celery in 2 different continents based on the soil compositions and stuff there, they might not actually be the same composition of molecules. Those might not even be the same at that level. So what I'm talking about for the purpose of this talk is, can I get to the individual molecules that are present in foods that might protect people from a given genetic disorder, right? So really, from the standpoint we're talking about, is diet is probably millions, if not billions, of different molecules, most of which have no impact, but some might, right? And then when I talk about microbiome, so I can't see the details of these slides to read them to you right now, but basically your microbiome, people might have read about this. You see it online every now and then. Diet and microbiome are linked to any disease that you could Google, right? If you Google it and diet or microbiome, you'll find someone who's claiming that that matters. But usually it's not clear what exactly is going on. It's very correlative, and they're not very sure. The reason for this is your microbiome, starting basically with your mouth, I guess, through your stomach and digestive tract, is comprised of many, if not trillions, of different bacteria, right? Or not different bacteria, but trillions of bacteria that comprise at least 500 species. These bacteria are actually mostly healthy, right? Like, you need them there. It would be worse off if they weren't there. And there's different types of species in different parts of your intestines, but there's a lot of them, and they're generally beneficial, right? And you want good ones there. But there's many, many different ones. And what microbiome I have and what anyone on this call have are probably different, for reasons that might be stochastic or genetic. Just what bacteria you get by chance might change, and they might change over the course of your own life, right? So what I'm talking about when I talk about microbiome, though, isn't even the collection of bacteria. It's the fact that they produce what is, by most scientific studies, considered an uncountable number of molecules, right? And ones that probably wouldn't even normally be in your diet. They also compete with you to eat the food you intake, and then they make their own set of molecules from the food you're eating, right? Those molecules are, again, billions, if not trillions, of potential molecules. 99.9% of them just don't matter from the standpoint of your health or your gut, but some of them could, in theory, explain some of what makes one individual healthier and one not. And so what my lab hypothesizes, the major overarching hypothesis of my lab, to give it a very simple schematic, is that for any given genetic background, there'll be interactions of your genetics with your diet and microbiome and those millions or billions or trillions of molecules that could, in principle, lead to more healthy or less healthy outcomes. And that might be common for certain genetic backgrounds, and it might be uncommon for things like Doug Whitney, where he has a very rare example of something that modifies it. Right? This is at least our hypothesis. Right now, there's no mechanistic data. I haven't shown any, and I don't think it's out there, but this is my lab's hypothesis, right? And so what we hope, though, is that if we could find the specific molecules that are present in a microbiome or diet that protect against disease, then we could utilize that to produce what I'm going to call Doug Whitney-like outcomes, right? Where we know someone should develop a disease, but we give them a specific thing that's going to protect them against that, right? That is the dream. And there's some evidence that this could be true. And so I'm going to give a scientific study. You don't have to read anything on the left there. It's just the title of this study. But really what this study was about is an example of a thing you're probably not going to associate with diet or microbiome. And this is ALS, right? So ALS is a neurodegenerative disorder. It's very, commonly studied here at VAI. It's one of our focus topics. And there's a gene called C9orf72 that is the major genetic cause of ALS in humans. And there was a model of this made in the lab, and they made it at Harvard, and they showed that, look, we have this model of ALS, and then they shipped it across the river to the place called the Broad, which is at MIT. It's only about 5-6 miles away. They re-derived the model, and then none of them got sick. And they're like, I'm not sure, is this a reproducibility issue, right? And I think people hear this in the news right now, reproducibility in science, because here you have 2 labs, and they have, one says this has ALS and we can use it. Another lab says there's nothing wrong with these animals at all. Like, there's nothing wrong with them. And what they did is they followed up on this, and they studied it for a couple of years. And what they realized is that if you took the microbiome out of one mouse and gave it to the other mouse, you could produce either outcome. You could actually completely protect them from a currently untreatable disease based on switching or eliminating their microbiome, right? And they highlight here in the text, in the discussion, I like to highlight this because it was so surprising to them that in one condition, you essentially had highly penetrant neurodegenerative symptoms, and another, they were essentially absent. And that was shocking, right? That the microbiome could have that big of an effect. And you don't see it in normal mice. You saw it only in this genetic model that otherwise you wouldn't have noticed, right? So in that context is where this was revealed. And so my lab's interested in this. And what we're really interested in, so I don't want to jump too far ahead with these images, is the challenge is 1, right? Challenge 1 is we assume that this is due to some molecule that's protective. And how do you find those, right? I just told you about how a diet is millions or trillions of molecules. Your microbiome is an uncountable number of additional molecules. And we assume that it's probably like 1 or a small number of them that's actually mediating any protection. And so how do you find them, right? This is actually the real challenge. You'll note that I just told you also about an ALS model. We still don't have any treatments for ALS. It's not like we made this discovery and now we're like, well, we'll just modify a microbiome and suddenly be able to treat ALS. And the reason for this is because even in that study, you don't know the species of bacteria. You don't know what molecules it's making. So you have an unknown mechanism of action with an unknown molecule, and it's very hard to turn that into a therapeutic, right? You just basically don't know what's going on. And so what my lab turns to is a very famous study at the time, but maybe not so well known now. And it's a study that used backyard soil microbes to transform human health. And so on the left, you'll see a picture of a guy named Selma Waksman. Selma Waksman is usually not known, even when I present this to scientists, even though he won a 1952 Nobel Prize in medicine. But he's famous in a field that most people know the discovery by a man named Alexander Fleming. And that was the discovery of antibiotics, right? So antibiotics were discovered a bit accidentally, right? Where bread mold that grew on a plate looked like it produced this new antibiotic that protect against antibiotic growth or bacterial growth. And Alexander Fleming sort of accidentally ushers in the age of antibiotics, which transformed human health. Maybe one of, if not the most important discoveries in the history of biomedical health is antibiotics. And what Selma Waksman did was actually only 7 years later, he made the hypothesis that this rare finding, this accidental thing, not unlike an accidental Doug Whitney discovery, is not uncommon. Basically, that this seemingly rare result he thought was actually probably common, and that bacteria might make lots of small molecules that are bioactive and could be used as antibiotics. And the most likely place for them was going to be in backyard soil. He was a soil microbiologist at Rutgers. And so he built what is now called the Waksman platform. And you can see a little diagram of it at the top here. But basically, he just collected dirt and grew the bacteria up and screened them in a high throughput approach using technologies only available in the 1940s, right? So nothing very complicated here. And he looked for antibiotics. And from this, he ushers in what's now known as the golden age of antibiotics. Either his lab or labs using this platform from the 1940s to the 1960s discovered every class of antibiotics we still use in the clinic today, except for 2 that were discovered later, basically transforming all of humanity. He wins the 1952 Nobel Prize for this Waksman platform. Nominally, it's for streptomycin, but this platform discovers almost all of the antibiotics that we still use today. And so the most audacious goal of my lab, if I'm to posit this, is we think bacteria also make lots of other molecules that that matter for biology. His platform could only find molecules from soil microbes that killed pathogens, right? Other bacteria, right? And that is great, but we think that there's also molecules that bacteria make, because it's the only way they can interact with their environment, that target animals, that sort of modify animal or eukaryotic physiology as it's known. And those would never have come out of this, but we want to build a platform that can find those, because we think those might be mediating who gets healthy or who doesn't in certain contexts of genetic disease. So that's our goal. But how are we going to find that? To do that, we need to have some high throughput system, not unlike Selma Waxman. So we need an animal model that we can grow at scales of millions or even hundreds of millions of animals. And this is where we turn to a really well-studied microscopic worm. You can't actually even see this with your eye, but it's all over the globe. It's called C. elegans, and it's been studied for 40 years plus now. It's been studies of this microscopic nematode, one of the best studied in biomedical research, underlie at least 3 different Nobel Prizes in medicine at this point. Lots of labs across the world study this nematode because it's got a lot of cool tricks about it. And my lab uses it too. And we think it's particularly useful here because we can grow lots and lots of these. They're not much bigger than the bacteria ultimately. They eat bacteria as their diet. They're smaller than you can see with your eye, and you can grow them germ-free. Basically, you can get rid of all bacteria around them by collecting their eggs. Their eggs are sterile. And then you can use this, and I'm not going to walk you through all of the platform. I will a little bit in the end, but what we built is basically a platform that looks a lot like the Waxman platform in the sense that you collect soil microbes, you array them into these plates, not unlike what he did, but we're going to instead use this new technology called CRISPR, which people might have heard of. CRISPR's in the news these days. It also won a recent Nobel Prize, but you can modify the genome of these little microscopic animals and create DNA variants that are the exact same ones found in human patients. So we take a human patient, we say they have this variant and it's causing this pathology. We're going to put that exact same variation into this nematode, right? And it will probably cause something somewhere similar or analogous, but then we're going to screen it at scale for a microbe that can protect it because we think those microbes exist. So that's what our goal is. And that, to put it back in the context I just showed you in the human, so just take out the humans and put in the worms. We think this is universal across biology, that for any given genetic background, it'll interact with a diet and microbiome. And for C. elegans, that's the same thing, right? They eat bacteria, and those bacteria go on to make up both their diet and microbiome. And those interactions between genetics and the diet microbiome could produce more healthy or less healthy outcomes. And this is what it looks like for a worm. And if this is true, then we can go after, with this platform, individual molecules. And so I'll walk you through a day in the life of my lab when we started this project. And so what you're looking at here is a compost bin in Rockford. So this is not far away. This is the backyard of my tech's father. It is a compost bin, right? Anyone has these compost bins, and we collect some of those, right? And then we grow them up as bacteria, right? And so we get a bunch of bacteria from a compost bin. We then need to make a model of a given pathology in C. elegans, right? And so here is an example where we took a juvenile onset Parkinson's disease. Like I said, VAI is very interested in neurodegenerative disorders. And so this is a gene that in humans causes a juvenile onset form of Parkinson's. There are no known treatments for this. And we basically make the exact same change in DNA in C. elegans. And I don't think you need to know much about C. elegans to look at these 2 pictures. On the left, you will see C. elegans as they normally grow. This is their lab diet. These are a bunch of healthy adult animals. And you'll see on the right that when they have this particular variation, that in certain contexts, they are not the left. They're not the picture on the left, right? They don't look so good. Actually, they sort of stop developing shortly after hatching, right? And so this is, from the standpoint of the worm, kind of analogous. Something's broken on the molecular scale that's the same as what we find in human patients. And so then we run it through our platform, right? We're like, are there any bacteria in the backyard that can make this animal healthier, that could produce a Doug Whitney-like outcome? And here's an example, right? So here is a Pseudomonas species. And I think even if you don't know much about worms, you can kind of appreciate that this has the same genetics as that picture in the middle, right? They are DNA-wise identical. They are siblings of each other, but in one microbial context, they're very healthy and can't make it to adulthood. And in another microbial context, they are more or less fine, right? They're like not 100% as healthy as wild type, but they make it to adulthood. They live normal lives. They reproduce, and it's more or less fine, showing, I think, how dramatic the effects of a dietary, because that's also their diet, or microbiome, we don't really know, right, can have. And this is just one pathology, right? But our hypothesis is that this is common, right? So here's another example. This is a variant in a mitochondrial fatty acid oxidation disorder. You don't need to know much about it, but it's commonly found in Japan. And in this context, we actually knew that variants in this gene sometimes result in very early lethality, right? In children under 3, and we have no way to prevent this. But in other humans, they'll have the same variant and live into their 60s with only mild pathologies. And so we knew that this doesn't make a lot of sense. So maybe this is one that might have this. And we here are 12 different bacteria, and you can see that actually we engineered the exact same variation. It's a specific change in one protein, this Y to D, which you don't need to know much about. We made the exact same thing in a worm, and in some bacterial contexts, they're very unhealthy and also show an early lethality, but in other contexts, they're actually completely fine. And this gives an example of the type of thing we're seeing. Here's a third one. This is the last one I'm going to show. We now have like 5 of these different models. This is insulin receptor. The one possible molecular biology gene that most people will have heard of is insulin. This is the insulin receptor. It's a genetic model of type A insulin resistance. In C. elegans, they enter this state of arrested development called Dower when they lose the insulin receptor. But here's a bacillus species that's also in the backyard where they grow up to adulthood semi-fine. And so we think that this is evidence that this type of phenomena is common, that we have many different genetic models that have no, well, other than insulin resistance, where there is actually quite good treatments out there these days with Ozempic and things, but most of them, they have no known treatments for these pathologies, and we put them in different environmental contexts, different dietary contexts, and we can find some that essentially give rise to healthy animals. And so the question 2, and I'm not going to go as deep into this, is, all right, well, you have a bacteria, but you just told me they make an unending number of molecules. How are you going to find the molecule? Like, you can't just like give humans this bacteria. And that's a great question, right? I mentioned this, that we still don't have any treatments for ALS, because if you don't know the mechanism or the molecules that cause it, you can't get a therapeutic. And that's sort of what plagues microbiome studies today. But the benefit of this system is that the diet and microbiome here has its own genome. Right? It's a bacteria, and therefore it's modifiable. And we can individually delete gene by gene in the bacteria and ask what in that bacteria is actually doing this. And so you can modify individual genes. And here's an example. We run these screens in reverse, and we look for mutant bacteria that essentially can't cure our model anymore. And here's an example of several genes in what's called the polyamine synthesis pathway, which you can see below. It doesn't really matter. They make metabolites, little small molecules that you can see below. And when you get rid of one of them, now the animals go the other way, right? So that bacterial gene, if you change it, the animals actually go the other direction. So now we're down to an individual gene in a bacteria that when that gene changes, it changes its metabolites, and that impacts the animal that eats it, right? So now we have an individual molecule. And in this particular case, because I don't want to go too deep into the science, we actually knew that the pathway you see below there mustn't be correct. It actually didn't make any sense with the genetics, and we spent the last 2 years working on this, and I'm going to skip to the end of the whole study, which is that we found by studying this gene in a bacteria that mediates an effect on an animal, this molecule that you see on the left, which is called N1-aminopropyl-augmentine. It's a non-canonical polyamine that's not normally found in any context, but we found that when bacteria make this, particularly if you delete that one gene, they'll make a lot of this. It actually inhibits mitochondrial function in the animals and causes them to stop growing. This is going to come out in Nature Comms in the next couple of weeks. And this is particularly exciting to us because here it proves that we can use our platform to find specific molecules that mediate an effect on animals. But it just so happened that a couple of years ago, people realized that deleting that gene in bacteria for reasons unknown was contributing to severe inflammatory bowel disease. And it wasn't exactly clear why deleting that gene in bacteria caused this. And what we think our study shows is that actually those bacteria are making this presumed previously unknown molecule, and that it's actually hurting the intestines. This is a study that was done, and we now think that this actually might happen in some people where their microbiome has lost that gene, because bacteria, there's about 1 million or more mutations in the bacteria in your genome per day, right? Bacteria are dividing all the time. Sometimes that goes wrong. And that maybe there are bacteria that produce this molecule and it actually contributes to IBD in humans. So we're collaborating with UMich West Hospitals. This is Dr. Michelle Musa-Moons to ask these questions of, things like, can we find this molecule actually in patients with IBD? And if so, we actually know how to go stop it. And so these are the types of things that come out of our studies. When we can get individual molecules, we can make specific hypotheses for, do those molecules cause disease, and can we block them if they're having a toxic effect, or can we isolate them and give them to people if they're protective? And so where my lab's going with this is we have some other molecules that seem to be active and mediate some of these effects. We have other conditions that we don't have molecules for yet, but we're hot on the trail of studying, and we hope to go forward with that. The last slide I have, because this is really ongoing research for me, is I just want to step back from sort of this technical science stuff and say that this project may or may not sound complicated to you. I don't know. But it's not. And I don't want it to seem like science is this hard thing that only a handful of people do, right? And so my lab, we work with Lowell High School for the last 3 years to show that even high school students, I think all these pictures I've showed you, anyone can look at these worms and see differences, and backyard bacteria are everywhere. For the last 3 years, students at Lowell have been going out into their backyards, collecting bacteria, and trying to do this in a high school with just the equipment that's present in a high school. I told you that we model our science actually on this phenomenal 1940s studies. Most high schools have all the things that you had in a lab in the 1940s. And I think they can do it. It's taken a little adapting, and we're working on it, but I am really passionate about this because when I was a student, science was kind of boring in the laboratory, right? You're just reproducing studies from 100 years ago, and you could either be wrong and fail at it or just reproduce something we already know. And that's not why most of us do science. Most of us do it because we could discover something. And I want to bring that experience to the high school. And they might not discover anything, they might, but they could, right? And so we think that this is doable, not just by us, but that this is a platform that anyone could take, at least with a modern lab, and build on for the genetic diseases they're interested in. So we're really trying to develop it for that lines and try to build it into curriculum to show students how science happens, that it's not just for a handful of people, that anyone can do this, given a platform, even people who aren't even 18 yet, right? So with that, I'd like to thank my lab. This is our funding sources. The NIH is critical to doing this research. I am a consultant for Novenensis, the Pew organization. And with that, my lab, who you can see there, who do all of the phenomenal work. And with that, I'm happy to take any questions.
Well, Dr. Burton, thank you for doing this. This is fascinating work. Mm-hmm. For those of you joining us, if you have a question that you would really love to ask, I think Dr. Burton is open to just about any questions, so go for it. Go ahead and use the email that you received with your reminder from Allison Baker at vai.org and submit that question, and we will try to get to as many of those as we can. So feel free to go ahead and just email those questions directly to Allison, and she will relay those to us. I want to get started. You are passionate about this work, and it's over my head, but at the same time, I feel like I learned something today. What drives you to do the work you do?
I mean, I am fascinated by treating and developing new treatments for disease. The reality, though, is like, why did I become a scientist? If you want to say, like, what drives you? I actually grew up, this is a random story, on a farm, growing up working on a farm in rural Wisconsin. I had no interactions with scientists. My parents didn't go to college. I didn't know what that was like, right? But I do know that when I was 16, there was a high voltage power lines behind the farm, and they grounded, and through a phenomenon called stray voltage, which I'd never heard of, electrical shocks were coming up out of the ground and shocking the cows. And this was a big problem for me because 25% of the herd got mastitis infections. There were birth defects. Like, it was a big problem. And it was such a big problem that there was lawsuits over it at the time that it's hitting other farms. And scientists from UW-Madison actually came out to try and figure out what was going on. And I don't know what happened. They probably didn't want to talk to some 16-year-old. But I asked them, did they find anything? And they told me that electric shock was bad for cows. And I was like, well, that is definitely easier than what I'm doing, because I could definitely have made that conclusion. And I was like, this seems like a lot easier job than farming. And at the time, I was like, I also really wanted to know. And so when I left, I actually went to Madison then 2 years later and signed up to be a dishwasher in the lab. Like, I just wanted to do it. I thought this looked like a great job. There was a question. Apparently, people weren't getting answers. And it snowballed into all of this career, right? And I love it, right? I love it. I think at the end of the day, I like just showing up for work and trying to find answers to things where there aren't answers, right? But especially in the context of if it can help people, I guess that's the real goal of what we should be doing, and it really does matter. And at the same time, I satisfy my need for discovery.
I love your curiosity. Okay, so let's talk about, based on the work you do, and I know I'm not asking for the magic bullet, but I kind of am.
Sure.
What should we, based on what you're learning, what should we be eating? What should we be growing possibly in our own backyards or not growing? What are some of those findings that may or may not be common things we all can do?
This is a great question. And one that I'm going to partially defer, right? I am not a clinician, and I do think that people should listen to their doctors on this stuff, right? Like, I think there's good advice there, and they're the people. What I will say, and what I really do want to stress, is that what our studies strongly suggest, and I think most studies do, is that what's good for one person might not be good for another person. That there's no such thing as universally good or maybe there's some things that are universally bad, but universally good is not necessarily a concept that biology is really into. Universality is just not one of those. And that what's good for one person might have no effect on another person or even be bad, and vice versa. And so to some extent, what we're learning is that we don't have enough information to answer that question, right? Is that it matters, that these details matter, that the nuance matters, and that if we don't figure out what's going on, we won't be able to answer that question. But by answering it, we might say, hey, you have these genetics. For you, this is going to be bad. Don't do this. But these things should be pretty good, right? And that answer might be different for someone else. And so I just think that the best answer I can give right now, today, is that we need more research, but specifically, we need research that's not correlative. We need research that actually finds the things, the individual molecules that matter or don't, so that we can answer that question, because it's an important question, right? And so it's a little bit of a defer, but I hope to, basically, that's the question we hope to answer. But
I wanted a shopping list, but that's okay.
Yeah, but I don't think the real take home might be that there's not a shopping list. So maybe don't buy in to too much of the trends. Is that there's some things, right? Like maybe don't go home and eat all day, eat Reese's as your only dietary source of calories. Like that, I mean, that might not be a universal only that food diet thing that would be good for you. Like diversity of foods is probably helpful. Past that, I think we need more research.
So Judy has sent in this question. I read that countries where children are in dirtier environments, they get fewer cases of type 1 diabetes. Is this related to the kind of research that you are working on?
It is not directly related to the research we're doing, although those studies are pretty convincing. There is a theory out there, right, that your body has an immune system, right? And that immune system wants to do something. And if it's real busy fighting off parasitic infections and things like this, it might maybe distract it from, if it doesn't have anything to do, it might attack self, right? And that balance is going to be stochastic and chaotic, right? Like sometimes things are just going to go wrong. And if in a perfectly sterile environment, the immune system might be like, well, maybe this is bad. I got to do something. And you might get higher incidences of autoimmune type pathologies like type 1 diabetes. It's not fully solved yet, but it does sort of look like there might be legs to that. It's not analogous to what we do because what we're doing, C. elegans doesn't have the same cells that would mediate type 1 diabetes. They wouldn't attack in that way. We're more interested in metabolites and metabolic diseases are our focus. But I think it's fascinating, right? I do think that this concept that, you know, your environment might affect your risk for disease is a universal principle. And so I'd say it's within the same general field, but not what my lab does.
Well, this is another question regarding that.
All right.
So this is from Marcia.
Yep.
And are there any applications that are taking place in your lab that connect to ovarian cancer?
Not again within my lab. C. elegans don't have ovaries. They do have oocytes. They form oocytes, but they don't have ovaries in the same way. So the reproduction strategies of microscopic organisms still involve sperm and eggs, but there's no analogous system. Also, C. elegans only live for about 3 days, so they don't really get cancer because they're not alive long enough for that to be a problem for them. So it's good for some studies. We are trying to make a cancer model in the lab. There are genetic variants, particularly in like KRAS, right, that can cause cancer to develop. And we would love, I actually got that exact question. Can you make this model apply to cancer? At an external scientific advisory board only 2 weeks ago, where experts in the field asked that exact same question. So it's a good question. We would love to develop it. It's not going to be ovarian specific, but we'd love to try and adapt this to cancer because what we know about cancer is that pretty much everyone has cells in their body that have all of these variations, but most times 99% they don't become cancer. And why is that, right? They have the variations, but they don't actually develop into cancer. And is there a contribution that we could identify that's protective or not or causative? I'd love to answer that question. We are trying to make a model of that, but it's very early days. And it's a little trickier, right?
So I know that you are studying soils from around Michigan, and you've said
Yes.
we have a lot to study. What are you finding in our soil that might be somewhat alarming?
We don't work on anything pathogenic, conveniently. There's nothing to me that is alarming in our soil. We pull things out of soil all the time. We do not in Michigan identify anything where I'm like, oh, that's surprising. I will say I did the studies in the UK a while back, and there's lots of species of bacteria out there, but there are certain classes where we'll just throw them in the autoclave immediately. And we would get occasionally there. We got one once where we just threw it away. And it might not have been anything, but we just get rid of it. We haven't found anything. Compost is largely non-threatening. I mean, if soil was threatening, you'd have a lot of problems, right? Like all of us see soil every day. Like my kids are out playing in the dirt all of the time. The soil is not really threatening. And we think we are starting to ask the question. I get this question to give a variant of it. You're like, well, why soil, right? Soil we're interested in because it has more bacteria than almost anywhere on Earth, right? There are more different types of bacteria there, and there's a lot of pressures for them to make molecules that interact with all these microscopic animals that are in their environment that might eat them. And they're kind of analogous. All animals are animals from the standpoint of a bacteria, and they make molecules to do this. And I do want to give people some examples that accidentally, there have been discoveries in soil microbes that have led to drugs. So rapamycin is sort of famously this transplant autoimmunity drug that is given to transplant patients. It's found in soil on Easter Island. It was an accidental discovery. And you can use it. It was probably something that fought with yeast. And you can use it all the way to the clinic. Ivermectin is sort of now famous and infamous. It was also identified from soil microbes to find in these large scale screens in Japan to find some sort of molecule that could kill parasitic nematodes because we wanted to fight these parasitic infections. These are molecules that are in soil bacteria that have been found before, probably because soil is such a complicated environment that they need to have them. I will say that I often get the question though, like, what about human microbiomes? Like, aren't you looking there? And we have just started to go down that route. It's a little more complicated to get human microbiomes, but we have just started to do so. And we're also going to look there. We think we can adapt our platform for that. Soil, nothing threatening, but lots of good stuff. And we are also moving into humans. Long-winded answer, but.
No, that's good. Jerry has asked this question. Can you talk a bit about gut bacteria changes over time? How many bacteria tend to disappear over a lifetime, and how many are new to the human organism after birth?
Well, look, it's a great place to start. Birth, right? I think one of the most fascinating things that I have ever read about science and the microbiome is that it's estimated, by some people anyway, that every human on Earth is colonized by E. coli within 20 minutes of birth, right? E. coli is, people might hear about E. coli infections, but that's like a very rare type of E. coli. Most E. coli is actually beneficial, and it's found in everyone's basically intestines. And it's so prevalent everywhere that no matter where you're born or how you're born, you'll find E. coli will find you within 20 minutes. Initially, early on in life, your gut is actually mostly aerobic, so it has oxygen. And so you're colonized by all these things that grow oxygen. Over time, as you age, those bacteria suck up all the oxygen, and then you start getting colonized by new bacteria that are anaerobic. So they grow in without oxygen, and those are different. But it probably, eventually you'll get some stable microbiome, right? Like there'll be some stability to it day to day, at least immediately, but lots of things can throw it off. And so you can temporarily go one way or the other. What I think is also fascinating about microbiomes is that it's not, though, just what you eat, right? Or even take. Like if you take probiotics, they pretty much don't colonize your microbiome. And the reason is that you have a lot of other bacteria there, and they don't want that competition. So they have good ways of just keeping everything moving along. So your resident microbiome is what it is. Over arcs of like a year or months, or if you do something very disruptive, you can see changes. That's not really my field. There are people doing that research of like what changes from what inputs, but it probably changes a lot. I guess over the life, all people start out with like E. coli and these oxygen aerobic bacteria early on. It'll go to an anaerobic environment. As you age, you'll get certain changes. Do those changes matter though? Is a real question. I think that's the question that everyone should really think about, is just because we see changes doesn't mean they do anything. Most changes probably won't on that scale. And so how do we pick apart which ones are good or bad or how from noise? Because bacteria are also just like living there and they might not do anything to you. Most of them won't. So it's a hard question to answer, but that's what my lab likes to do. Which ones matter? There's changes. There's lots of changes. Change all the time. Which ones matter?
That's a great way to look at it. I want to know what's next for your research.
Well, I think right now we are focused on these 5 disease models we have, where we have microbes that make them healthier, and mechanistically finding, some of them we have some molecules for, but other ones we don't, and just showing that this works. And in the future, we'd like to take it out of worms, right? Like the next goal is to say, all right, that works in your context, but is it specific? We think the whole argument is that this is not unique to our worm model, that this would be true in any model. And the long-term goal is to translate that away so that one day we could get to an intervention in the clinic for these same pathologies. So I guess what's next is to find the molecules for the ones we already have, and then ask, would any of these potentially work to be one day in the clinic? And that's our goal.
Why worms?
Because to do this, to get mechanism, you need to be able to say this is the bacteria and this is the gene. And that's hard to do in almost every animal, because almost every animal has microbiomes comprised of thousands of species of bacteria that you can't control. Worms are great because they grow fast, you can grow them at scale, but really the key is that I can get 100,000 or 1 million animals that are germ-free, so they have no bacteria, and I can put that model in the presence of only one bacteria at a time, right? That is absolutely fundamental to being able to do this, and they're one of the only animals that lets you do that. And so we use worms because to run these high throughput screens, this isn't something you could do with anything else. And so the ability to get lots and lots of germ-free animals with tools that are well established because it's been studied for so long is the reason.
So worms are your thing. All right.
I mean, for now.
I'm very curious. You have studied around the globe. You have taught at prestigious places like Cambridge University. Why Grand Rapids, Michigan? Why Van Andel Institute?
Well, I mean, I am from the Midwest originally, so this is a big point. When me and my wife were looking for places, we were over in England, the Midwest was our #1 choice because it was close to family. And we had just had a kid, and that was what we were doing. It was also, that time was 2020. And as you might remember, people on this call, that was a time where there was a lot of changes happening at the time, and there wasn't necessarily a lot of places were sort of reserved on hiring for faculty jobs, but Van Andel Institute really was like, there's a lot of talented scientists out there. The jobs have sort of dried up temporarily because everyone's on pause. Let's put out a big search, right, for a number of junior faculty. And this got passed along to me, and I was like, oh, Grand Rapids, it's pretty close to home. And I read more about it, and they have just an absolutely excellent setup here to do science. And my background is actually on how a mother's environment affects offspring. A lot of my background is in that field. And I was starting this new bacteria project. At the time, it was brand new. And I was like, hey, I want to do both of these things. Can I do both of them? And it was the only place where, like, we will help you do both of these, even though that one's, at the time, we had no data showing that would work. It was kind of a moonshot idea. And they're like, we'll take a chance on it. And I was like, that's amazing. Like, nowhere does this. This is a really, really great setup. And so it's a really attractive place to move. I love the Midwest. I'm excited to be back. And it's been everything it was built to be, right? It's been a really great setup.
Well, we are thrilled to have you. My final question for everyone who has been joining us today: what gives you hope in the work that you do every day?
It gives me hope to see that we can take an animal that should get sick and we can make it healthier. And that gives me hope that we can do that's a true thing that we can do. It might not be easy to adapt that all the way to the clinic, but the fact that we can do it at all. We have one model where, these animals just lay eggs, but none of the eggs ever hatch, right? And I guess, if you're getting chickens or something, that's fine. But here we know that it's because of this metabolic defect. And we even have a diet / microbiome where we give it. And this animal that never gives rise to eggs, they hatch and grow up. And that is fascinating because that was thought to be unachievable. And just with a dietary intervention, I hope we can do that one day, right? I hope that works. And we're positioned right next to Helen DeVos Children's Hospital. I hope to collaborate with them on some of these things because that's what we care about.
I love it. Thank you so much for giving us your time and your expertise today. This has been wonderful. If we were all in a room together, I would say, let's give him a big round of applause. I'm doing it for you. Thank you so much.
Thanks very much. It was nice to be able to talk with people and great talking with you again.
And friends, if you are joining us and you're saying, I'd love to learn more about the work he is doing and more about the work taking place at Van Andel Institute, you can head to our website, vai.org, and learn more about it. You can also get on the mailing list to find out about other events similar to this. Follow us on social media. You'll be in the loop on everything. Now, we have one more public lecture series. It's taking place on December 17th, and it actually involves something that Dr. Burton did mention. The public lecture series is called The Promise of Gene Editing Using CRISPR. What is it? How does it work? And how will it impact you? Sign up now. Go ahead and plan to join us on December 17th for that conversation. Thank you so much, and I hope you have a wonderful afternoon where you live.